So the title of this paper and today's talk is Positive and Negative Chemotaxis of Enzyme-Coated Liposomes. So the primary objective of this work is to try and understand how can we steer soft colloids or protocells in both positive and negative chemotactic direction. So as we all know, directional migration is crucial for the survival of living systems. It enables cells to move towards food, away from toxins, coordinate collective behavior, and transport cargo. Here is an example of a vesicle that is being transported across a cell through the conversion of chemical energy from ATP hydrolysis into mechanical motion. So here is a second example where a white blood cell is chasing a foreign particle by sensing it in its environment, moving towards it, and ultimately engulfing it. So this kind of movement due to chemical signals is called chemotaxis. So chemotaxis is generally seen in two directions. When the cell moves up the concentration gradient or towards higher concentration of chemical, it is called positive chemotaxis. And when the cell moves away from high concentration or down the gradient, it is called negative chemotaxis. So for a very long time, it was assumed that only motor proteins in cells or in living systems are responsible for directional motion. It was about a decade ago that it was found out that even enzymes, traditional enzymes that just catalyze reaction, are able to move directionally when placed in gradients of their reactant. So further studies with these enzymes when they're attached onto polystyrene beads or encapsulated inside polymer beads are able to show directional motion. So these active systems that are created synthetically in labs are called micro or nanomotors. Micro and nano because of the size or length scale in which they are found in, and motor because of their ability to convert energy from one form to another, similar to a motor in a car. So one similarity that I want to point out among the four studies here is that all of them show positive chemotactic motion, that is towards higher concentration of reactant. Negative chemotactic migration in enzymatic systems has not been shown. And also one more thing is that the motion that arises from enzymes associated with natural membranes, natural materials such as lipid membranes is unknown. So liposomes or other soft colloids have recently come under the spotlight as potential motors due to their high biocompatibility and bioavailability. Because of these characteristics, they have the potential to be cargo delivery or drug delivery vehicles within the body. Also, because of their structural similarity with membranes of cells, they are able to be used as artificial cell models to study chemical and biochemical reactions in vitro. So these liposome structures are easily formed in the lab by the self-assembly of phospholipid molecules. They're able to self-assemble to form an enclosed volume, an aqueous enclosed volume, and they are able to be easily functionalized on the surface to have different functionalities such as enzymes or charges or proteins and so on. So in this study, what we wanted to do is to see if liposomes are able to move directionally when enzymes are attached onto their surface. And if so, we wanted to see in what direction do these enzyme-coated liposomes move. So as I mentioned earlier, enzyme molecules as well as enzyme-coated particles are able to show positive chemotaxis. So we hypothesized that these enzyme-coated liposomes would also show self-generated positive chemotaxis upon catalysis. So in order to test this hypothesis, we mainly employed three techniques. The first technique is thin film hydration technique to form the liposomes. And then we extrude them using 100 nanometer polycarbonate membranes to form uniformly sized liposomes. And this is how they look under the cryo-TEM microscope. So these lipids that I show you here are the main lipids that we use in this study. L-alpha phosphatidylcholine represents the backbone lipid that makes up most of the liposomes, We add a biotin lipid so that we can attach enzymes onto the external surface. And rhodamine lipid gives fluorescence characteristic to the liposome so that we're able to see them under the confocal fluorescence microscope. And once we form these uniformly sized liposomes, we want to attach enzymes onto their surface. And we do this using the biotin-streptavidin conjugation technique. The enzyme is first biotin-lated, and then streptavidin is attached onto it, and then they are mixed with the extruded liposomes in order for the enzymes to attach onto the surface. So when we look at these enzymes under DLS, we are able to see a size increase that indicates that enzymes are attached onto the surface. We also looked at these liposomes under cryo-TEM as well as SDS page to confirm that enzyme is indeed attached onto the surface of the liposome. So once we form these active systems or enzyme-coated liposomes, what we wanted to see is how they would move in imposed gradients of solute or reactant. So for that, we use a 3-inlet, 1-outlet microfluidic channel. Typically, we flow the liposomes through the mid channel and buffer in the side channel. This is the control. When we replace one of the side channels with reactant or solute, that constitutes the experiment. So we compare the control and the experiment to determine how much the lipid vesicles or liposomes have moved in the gradient. So here I want to mention that this 3-inlet, 1-outlet channel is connected upstream to a syringe pump, which is able to control the exact flow rate in the channel. So controlling the flow rate enables us to exactly have the correct time for interaction between the buffer and the lipid vesicles. So the flow direction is from left to right, and we looked at how the liposomes have moved perpendicular to the direction of flow. So here is a typical graph that we get from analysis of fluorescence intensity across the channel. And the left part of this figure corresponds to the top channel, the middle part corresponds to the mid channel, and the right side corresponds to the bottom channel. So since the liposomes are fluorescently tagged and flown through the middle channel, we can see that the fluorescence intensity is highest in the mid channel, and it goes down to 0 in the side channels as the liposomes move into the side channel through Fickian diffusion. So the first enzyme that we tested is catalase. Catalase is a relatively fast enzyme that converts hydrogen peroxide into water and oxygen. So similar to the previous studies where catalase molecules as well as catalase-coated particles show positive chemotaxis, we also see that the catalase-coated liposomes are able to move towards higher concentration of hydrogen peroxide. So the next enzyme that we looked at was urease. Now, urease molecules as well as urease-attached particles also show positive chemotaxis. So what was very surprising to us is that when urease-coated liposomes are placed in gradients of reactant urea, they show negative chemotaxis. Now, we also tested the liposome movement without enzymes on them in gradients of urea, and we see that there is no chemotaxis or negligible chemotaxis. So this led us to suspect that the products of the urease reaction is what is causing this negative chemotaxis. And so that is exactly what we tested next. We looked at inactive liposomes or liposomes that do not have enzymes on their surface in gradients of the product of urease reaction. And indeed, we see that the liposomes move in the negative chemotactic direction, that is away from high concentration of ammonium carbonate. So not only does ammonium carbonate make the liposomes show negative chemotaxis, we also found that there is a number of solutes that make the liposomes show negative chemotaxis. So this led us to suspect that there is an additional mechanism of movement acting on these soft liposomal particles and making them move away from high concentration of solute. And that brings me to the next part of my talk, where we try to find out what exactly is the mechanism of negative chemotaxis in inactive liposomes. So for the rest of the talk, I'm going to concentrate on inactive liposomes in gradients of solute. And to answer this question, we had a number of hypotheses that are listed here. So I'm going to go through them one by one and show you that we were able to rule out 1 to 3. And I'll show you currently how our standing hypothesis of liposome movement according to the Hofmeister series of ions. Okay, so the first mechanism that we looked at was diffusiophoresis. So diffusiophoresis is the movement of a particle when placed in gradients of electrolyte or non-electrolyte. So today I'm going to focus on electrolyte diffusiophoresis. So electrolytes such as salts have anions and cations. And due to the difference in diffusivities of anions and cations, a spontaneous electric gradient is able to be formed in solution. So a charged particle, when placed in gradients of such electrolytes, is able to move directionally one way or the other. Now, how the electric field is set up depends on the charge that is present on the difference in diffusivity factor as given here. So a positive beta value would have an electric field facing one way, and a negative beta value would give you an electric field facing the other way. So in order to test whether there is diffusiophoresis in our system, what we did is we subjected the liposomes to gradients of beta positive and beta negative salt. So here, ammonium bicarbonate makes the liposomes move away from high concentration. But when we tested it with a beta negative, the opposite charged beta value salt, NaCl, we see that there is no movement. So from these experiments, it tells us that diffusiophoresis is probably not a dominant mechanism in our system. So the next hypothesis that we had was that if there is any density-driven flows in our system. So solutal buoyancy differences between reactant and product has been shown to move fluid in enzyme micropump systems. So in order to eliminate any such effects in our system, we looked at the movement of solid polystyrene particles in gradients of reactant and product of the urease reaction. So the reason we use polystyrene beads is they are denser than liposomes, and if there were any density-driven flows, the effect that the particles would feel would be much more pronounced. But when we pass these 200 nanometer particles in gradients of urea or ammonium bicarbonate, we see that there is no movement. And so we can effectively rule out density-driven flows in our system. So the next hypothesis that we had in mind was osmophoresis. Osmophoresis is the mechanism by which a membrane-bound colloid is able to move in a gradient of impermeable solute. So in this situation, what happens is that due to the osmotic pressure difference inside and outside the vesicle expels water from the side facing the higher solute concentration and admits it on the opposite side, thus propelling the vesicle away from high concentration of solute. So in order to see if osmophoresis is dominant in our system, we looked at our experimental negative chemotaxis values and compared it with theory. And here what we observed is that we were able to get an overlap only if we had a huge fudge factor of 10 to the power 4. So our experimental values were 4 orders of magnitude larger than what was predicted by osmophoresis theory. So this tells us that osmophoresis is also negligible in our system. So at this point, we had gone through several hypotheses that were available in literature, and we were not sure what's going on in our system. So we went back to literature to dig more about what could be happening, and that's when we came across the Hofmeister series. The Hofmeister series is a series of anions or cations according to their ability to salt out or salt in organic biomolecules. Now, salting out means that the salts are able to promote aggregation of organic and biomolecules in solution, while salting in means that they are able to promote solubilization of the organic and biomolecules in solution. So the Hofmeister series has been around since the 1800s, but it is not completely understood how it actually works. But a lot of research has been done, and they show that it could be due to a combination of factors such as shape, size, hydration thermodynamics, charge distribution, hydrogen bonding capability, and hydrophobicity of the ions itself. So when you look at the Hofmeister series, when you go from left to right, we go from well-hydrated salts to poorly hydrated salts. Well-hydrated salts have a good water structure around them and do not associate well with hydrophobic parts of organic or biomolecules. Whereas the poorly hydrated salts have a poorer water structure and are able to associate with organic biomolecules and be attracted towards them. What we observed in our experiments is that the salts that induce negative chemotaxis lies in the salting out region of the Hofmeister series. So in order to test the hypothesis that liposomes indeed move according to the Hofmeister series, we picked salts from the other end of the series. And indeed, what we see is that the liposomes are showing a reverse positive chemotactic effect with salting in salts. So salts that have a good water structure around them, such as bicarbonate, they have a good water structure around them and they are depleted from the lipid-water interface, thus repelling the liposomes and making the liposomes go in the negative chemotactic direction. Whereas salts such as thiocyanate have a poorer water structure around them and are able to be enriched at the lipid-water interface, propelling the vesicles towards higher concentration of the salt. So we believe that ion enrichment or depletion from the surface of the lipid-water interface makes it go in the positive or negative chemotactic direction. And so with that, I'd like to conclude. We have shown for the first time that active and inactive liposomes are able to move in both positive and negative chemotactic direction. We have successfully ruled out diffusiophoretic, osmophoretic, and density-driven transport mechanisms for the movement of liposomes. We show that the liposomes are able to move in accordance with the Hofmeister series of ions. And this is the first step in programming directional motion to form adaptive life-like materials. And so with that, I'd like to thank my advisors, Dr. Ayusman Sen and Dr. Daryl Velagol, and my co-workers and collaborators on this project, Drs. Shubhadeep Ghosh, Farzad Mohajarani, Paul Kremer, Ting Lu, and Linisha, and also the members of Sen and Velagol Lab. And thank you, Nanomotor Update, for inviting me for this talk. And with that, I'd be happy to take any questions.
Okay, Ambika, thank you for your wonderful talk. Now we move to the Q&A session. Anyone from the audience who have a question, just unmute yourself and speak loud, or you can text in the chat box.
Hi, Ambika, this is Tiger.
Hi, Tiger, how are you?
Good, good. Thank you for the wonderful talk. I really enjoyed it. I had the feeling that I was watching a movie, a suspense movie, right? You had the very nice observation that you try to unravel the mechanism by eliminating suspect one by one. And I really appreciate this kind of approach. And this is kind of approach that our lab is trying to do our research as well. So if I can comment as someone who just watched the movie, I like to say that the movie ends a little bit abruptly, especially the second to last the slide, where you mentioned the mechanism of how the ion accumulation and depletion induces chemotaxis. Can you perhaps elaborate on that part and try to connect the ion accumulation depletion to exactly how it moves the liposome?
Okay, sure, yeah. So to be completely honest, we are also not sure what exactly is happening. So this is something that we saw that we're able to see negative chemotaxis and positive chemotaxis according to the ions in the Hofmeister series.
Yeah.
So when we look back at exactly what is happening at these ions, as I mentioned, there are a number of factors that could be affecting this here. So what we chose to do is we looked at the hydrogen hydration or water structure around these ions, and we observed that poorer water structure, poorer water structure, which is like thiocyanate and perchlorate, are able to be attracted to the lipid-water interface. So this could be because that the liposome itself has a hydrophobic part, and because these are poorly, the structure of water is poor around these ions, they are able to interact with the hydrophobic parts of the liposomes. Whereas in carbonate, the water structure is really strong and very ordered, that it does not want to break that water structure to interact with anything. And so that's why we think that it is repelled from the lipid-water interface.
Right. So, okay, so the last part of your last sentence, right? It is repelled from the liposome its surface. What's the mechanism behind this repulsion? I understand some ions like the interface, some ions do not like the interface, but why does it, where does the force come from?
That's a good question. I'm not 100% sure. It could be something to do with like osmotic gradients,
Ambika,
maybe pressure gradients.
I have a hypothesis. This is fundamentally, this is the same as diffusiophoresis. This is just a different way for diffusiophoresis to happen, right? Because in the theory of diffusiophoresis, you have ions who are interacting with a colloidal surface, right? And that interaction potential will give you some sort of, you know, if you put that interaction potential in your equation, you get some sort of slip velocity, right? And
that's correct.
Right. So here, instead of the regular interaction, you have this sort of a solvation-induced interaction, right? Some ions do not like water, so that they actually interact with the liposome surface more. This is as much I can say, but I have a feeling that this is along the same line as diffusiophoresis.
Right. So in diffusiophoresis itself, you have the electrophoretic component and then the pressure-driven component. So I guess like when you say it's solvation, maybe the pressure-driven component is having some sort of dominance, and that could be what is happening. Yes, that's absolutely correct.
Exactly for the neutral diffusiophoresis, right?
Yes,
because there's no electric, right? There's no electric component in that. So everything that's happening is some sort of a solid particle interaction, right? And so perhaps this this is another way of that happening.
Yeah, that's correct. Yeah.
Really interesting, fascinating. Thank you so much.
Thank you for your questions. They're very thought-provoking.
Any question? Okay. Ambika, can you see the question from the chat box?
Oh, yes. Yeah.
Okay.
Have you tried these experiments without microfluidic channel? Oh, that's a great question. I actually have not tried them outside the microfluidic channel. The reason for this is these liposomes are actually, they're very difficult to look at if they're not, controlled by some external flow. So if we just put them in a Petri dish, it's really difficult to visualize them under the microscope because they keep going up and down the frame. But yeah, the shear question, actually currently in our lab, there is some students who are trying to make a shear-free microfluidic channel. And it would be really interesting to see if, like, without shear, how these would move just in gradients of solutes.
Well, Ambika, I'd like to follow up with Lei Wang's question, because one of the main feature of a liposome is it's soft, right? But
Is what?
It's soft. It can deform.
Yes.
But I don't think you, in any part of your talk or your argument, the softness comes into play. Am I right?
Yes. So
then there are two questions. One, if you somehow coat the liposome or the main component liposome onto a solid particle, you would in principle see the same thing, right?
Yes.
And the other thing is, because it's soft and it can deform, any sort of a pressure distribution will cause the particle to deform, right? Yeah. And have you ever seen that?
That's a great question. So let me answer the soft part first. So our system of liposomes are 100 nanometer in size. And so since the size is so small, there is negligible deformation in our system. But then if you make them bigger, maybe go to the micron length scale, 1 or 10 microns, you would definitely see those deformations, but we have not looked at that. So for our experiments, we considered these liposomes to be non-deformative because of their size, their small size and high curvature. So that is something we have not looked at. And your second question about coating particles with lipids, we have indeed seen that. So what we did was we took 120 nanometer silica beads and we made liposomes lipids to assemble at the interface, and we looked at them in gradients of ammonium carbonate and sodium thiocyanate, and indeed we see them show the same behavior. Awesome. So yeah.
Perhaps this is a research direction for the future, right? Because we don't see a lot of liposome-based motors in the literature. Mostly our motors are solid particles, and but because everything happens on the surface, and and those things induce force, and so if you have a force acting on a soft body, it will deform. And that might change the motor dynamic dramatically, and even couple back to the reaction, so you have some sort of nonlinear system. So liposome-based motors, I don't know, Ambika, have you seen a lot of those in literature? What's your thought?
So liposome-based motors, like you mentioned, it's not very common, but it does have the added advantage because, like you said, if we are controlling the motion with enzymes on the surface, we can definitely have, like, cargo that can be inserted inside the vesicle. So it can be like, we can use these enzyme-coated liposomes to move cargo around. So definitely that is one additional direction to look at. And also we can control the different chemistries of the liposome itself by adding charges. You can add like charged lipids, you can add cholesterol to make it stiffer, or you can add like oleic acid to make it more permeable. And maybe like, when it reacts with the urease or something like that, you can have some sort of deformation that would enable the cargo to be released from inside or something like that. Yes.
Cool. Thank you, Ambika.
Yeah. Okay, did you see the influence of different ions on? Okay, so influence of different ions on motion speed. So we have not looked at all the salts. We have only looked at the salts that were relevant to the enzyme itself. So for example, urease converts urea into ammonium and carbonate. So we tested the movement of urease-coated liposomes in gradients of ammonium carbonate, and indeed we see it move the negative chemotactic in the negative chemotactic direction. But one thing that I want to mention that was very interesting, let me see if I can pull up the slide, is that, yeah, here. So here we see the movement of active liposomes in gradients of urea, and you can kind of see that with the concentration increase with urea, it kind of plateaus. But the same behavior when we test it in ammonium carbonate, it kind of goes negative all the way. There's kind of no plateau. So this is also another indicator that the product is indeed interacting with the liposome. And in the case where there is no urease reaction, it just interacts with ammonium carbonate. So here there's kind of like a dual, two kind of interactions going on. One is the enzymatic reaction and the reaction of the phospholipid with the ammonium carbonate. So yes, there would be some effects, and they might change the properties of the enzyme. Yeah, they might change the properties of the enzyme, yes. In the end, does the enzyme work to enhance negative chemotaxis or the motion of active? So I'm not sure if I understand this question properly. I wonder in the end, does the enzyme work to enhance negative chemotaxis motion or not? So the enzyme along with the phospholipid enhances the negative chemotaxis. This is because if you have just enzyme molecules, urease would actually show positive chemotaxis. And urease enzyme, when attached onto polystyrene beads, shows just positive chemotaxis. So there is an interplay of enzymatic reaction as well as the solute phospholipid interaction that makes it move in the negative chemotactic direction. And it can be applied to both active and inactive cases, because in the active case, it's kind of a self-generated negative chemotaxis, whereas in the inactive case, it's just responding to the gradient of salt. I hope I answered that question. Okay. Oh yeah, that's right. Tanya's work on enzyme liposomes. And I think they attached some, they have some cargo inside it as well.
Hi. Well, we had the enzymes inside. Then,
okay.
Yeah, and then of course we need to look for a strategy to permeabilize the membrane and allow the exchange of products, because in your case you have it outside. When we put the enzyme inside, we also observed that it's difficult to have this exchange, so we didn't observe motion until we came up with a strategy to make some pores in the membrane, and that was the challenge if we want to place them inside, just to add to the discussion.
Thank you.
Any questions from the audience?
Oh, I think I see someone has a raised hand.
Well, I think he raised hand in the last minute talk. Okay.
Oh, okay.
Okay. Let's wait for another 10 seconds. If there are no more questions, thank you, Ambika, for your wonderful talk. And this video will later be uploaded on the YouTube.